Hi, my name is Neel Moudgal. I'm a senior at Saline High School in Saline, Michigan, and today I'd like to tell you about my project, which focused on using unassigned NMR chemical shifts to model RNA secondary structure. So RNA, like DNA, is part of a class of molecules called nucleic acids that store genetic information in the form of base pairs and base sequences. RNA was thought to be a simple messenger molecule which took information from the DNA in the nucleus of a cell and moved it outside to the cytoplasm where it could be made into proteins. However, more recent studies show that RNA performs a much wider set of cellular functions, including catalyzing reactions, regulating gene expression, and turning on and off the production of cellular metabolites. As a result, it has become increasingly important to understand both what functions RNA molecules have in a cell and how they perform these functions. And as it is in a lot of concepts in biology, for RNA, structure determines function. Therefore, in order to have a complete view of RNA's function, we must first begin to understand RNA structure and the reasons behind why it structures the way it does. Multiple approaches for this have been proposed, the most famous of which is a minimum free energy approach, which chooses the structure that has the lowest energy, as this is usually the most stable one. However, this suffers some drawbacks, including the fact that not every molecule actually assumes a thermodynamically favorable state, and its accuracy is often inversely proportional with length of the molecule, so it therefore fails for large molecules. My project focused on rectifying this problem by using both experimental data along with simulated data from an ensemble based on thermodynamic considerations. So we took and for a given RNA molecule, we took the segment and determined with a set of low energy structures, not necessarily only the lowest energy, but a set of 500 or so structures that have lower energy than other possible structures, as these are likely to be better candidates. From that, we used a model that'll predict a type of data called chemical shifts. Now, chemical shifts are an experimental data point that's obtained through a method called nuclear magnetic resonance spectroscopy. And we went the other way in this sense that we took a simulated model and tried to predict what chemical shifts we would see if we had actually used that molecule. We also took a set of chemical shifts from an experimental data, and we're able to compare the predicted chemical shifts from our different candidate molecules with the chemical shifts from experiment, and with those comparisons, we were able to assign weights to each of the candidate molecules. Each weight corresponds roughly to the probability with which each molecule would be sampled. The highest weight is called the dominant structure, as it is the structure that's most likely to be sampled. Our study then looked at the ensemble average properties, where we take the weighted average across these different weights of the different simulated molecules as metrics for how our prediction would be. And we found that our model predicted with comparatively high accuracy the structure of RNA and did very well for large RNA segments. For example, a graduate student in the lab in which I worked tested my model on the 3' and 5' untranslated regions of the SARS-CoV-2 viral genome, and found that the model predicted the DMS-derived reference structure with high accuracy and at times better accuracy than the minimum free energy method. As a result, we were able to conclude that this model can be used in both theory and application as a way to inform researchers about the structure of RNA molecules. Our hope is that this research can be used to help inform drug discovery and allow physicians and scientists to understand the different mechanisms by which diseases and RNA interact, and hopefully through these find cures for a variety of diseases that plague humanity.